For exercise 12, heredity, we're going to actually go through not only some basic concepts about, when we're talking about genetics, right? What are genotype or phenotype, but also how we can actually, based on a parent's kind of genetic makeup and decide what's the offspring's genetic makeup, genotypes of the offspring, as well as phenotype of the offspring. Well, depending on what kind of the relationship, right, between the alleles, that could be slightly different. But basically, the whole exercise 12 is exercise practice for you, right? Giving you a specific example and having you work out, figure out how to do that, how to tell what would be the offspring's genotype and phenotype. So in the lecture, I'm going to walk you through how I'm going to do that. And but then the examples I gave you in the lecture may be different from what you have to answer. You have to, practice in the exercise 12. But that's the purpose, right? I'll give you some examples, but for you to really know whether to say whether you really understand about it, you actually have to do that yourself. Okay, let's go ahead and start with some basic concepts. So genotype, what is genotype? That's the genetic makeup. Okay, then phenotype basically means the physical expression of the trait, right? So sometimes the genetic makeup and the physical expression of the trait is straightforward. Sometimes it's not as straightforward. So we are going to see depending on what kind of situation, then how do you going to decide, right? Based on the genotype, then decide figure out what's the phenotype, what is the expression, what kind of trait is going to be expressed for a particular genotype. And how are we going to do that? Because this is kind of this is a karyotype. Basically, it's the 23 chromosomes in our human, right? You can see them paired together because we have the two chromosomes paired together, because for each chromosome 8, we have 2 copies of it, right? So those 2 are called homologous chromosomes, right? So 2 copies of them. And so essentially, for each gene, right, the genes are the genetic materials encoding for protein. You can find them in the chromosomes. You have for each gene, you also have 2 copies of the genes, right? One on each of the chromosomes, homologous chromosomes. And so then not all the genes are exactly the same, right? For example, gene A on this chromosome and gene A on the the homologous chromosome, they may be slightly different, even though they encode the same protein. But then this protein could be slightly different, so therefore the function could be slightly different. And so that's what we call alleles. Okay, so there are variations of one gene. So you essentially have always carries 2 alleles for each gene, because one on each one of the homologous chromosome, right? So depending on the allele, we have different type of alleles. Some of them are called dominant allele. That means if you express this variant of the gene, so the physical trait or the expression of that gene is dominant. So that means you always see the expression of that gene. Sometimes the allele is recessive. Recessive means is the physical expression of that trait is masked. It tends to be masked by dominant allele. but there are times the recessive allele you are going to see the trait of it, physical expression of the trait, when you have just 2 copies of the recessive allele. There's nothing to mask it, then you will have the expression of the recessive allele. Okay, so then after you understand what is alleles, right, then you can see, well, when we describe a person, then we could say it's homozygous. That means if it has 2 identical alleles. Okay, it could be 2 identical dominant alleles, could be 2 identical recessive alleles, but either way, it's homozygous, meaning it's identical, 2 identical alleles. Heterozygous meaning a person with 2 variant alleles. That means the 2 alleles, 2 copies in gene it carries actually are 2 variants. So then what's this person's phenotype is going to depending on the particular alleles it carries, that this person carries. Okay, so let's look at the example, right? The most commonly, we are going to start with the most straightforward kind of classification of alleles. That would be means the dominant, we talk about dominant and recessive allele. Right, so let's look at example with the dominant, straight dominant, and recessive allele. So dominant allele we usually represented by a capital letter. So this particular trait, physical trait we're looking at is widow's peak. So you have a peaked, a curved hairline, or you have a flat hairline, a straight hairline, right? So the widow's peak is actually a dominant allele. So that means it's going to be represented by a capital W. That means if you carries, if you only carries one, even if you only carries only one widow's peak allele, the big W, then you will present with widow's peak. Okay, and then the recessive allele, that means it tends to be masked, right? But so it is represented by a lowercase letter. So in this case, it would be the recessive allele is presented like this, a lowercase w. And we said, well, normally it's going to be masked, right, by a dominant allele. But if you have 2 copies of the lowercase w, then the physical trait is going to be presented. And then that would be the straight hairline. Okay, so it's only going to be presented when you have a homozygous lowercase w. And how about, so if the individual is homozygous with capital W, obviously it's going to have a phenotype. So this is the genotype, right? And then the phenotype would be widow's peak, right? But then how about the heterozygous? That means this individual carries a copy of the dominant allele, a copy of the recessive allele. Well, because it carries a copy of the dominant allele, then the dominant allele trait is going to be presented. It's going to show then that would be widow's peak. So 2 heterozygous here, right? Both are actually going to show the widow's peak. Only the homozygous recessive allele shows the recessive trait, that will be straight hairline. Okay, so you're going to say, wow, why they're, put in this kind of squares? Well, this is 2x2 tables. This is used to actually figure out from the mom and dad, the mother and the father. So if the mother, in this case, is showing the mother carries a W, a capital W, a dominant allele, also carries a recessive allele, right? And then the mother, so then you can see it's showing you in the form of the oocyte, secondary oocyte, and associated cells, right? Because then this mom, the gametes from this mom, then it's going to carry either, right? Because then with the gametes, it only carries one allele. right? And then it's going to carry either the 50% of the oocyte is going to carry the dominant allele and carry the recessive allele, right? Because it only has one copy. And then how about the dad? If the dad also has is heterozygous, carries dominant allele as well as recessive allele, is the sperms from the dad is going to carry only one of those two alleles. So 50% of them going to be big W, 50% of them going to be lowercase w. And so that's how you write it up, right? So you have the mom's genotypes, right, on top, right? And then the dad's genotype on the first column here. Okay, after you write out the mom's and dad's, right, gametes here, then you can start to work on the Punnett square that's generated, right, using the genotypes of the mom and dad. Right? For the first scenario you're looking at, okay, you could be getting a capital W from the mom, and then a capital W, right, the dominant allele also from the dad. So when you have this situation, then you'll get homozygous dominant allele, big W. And then of course, then if you look at this, then you are going to have a possibility to get recessive allele, lowercase w, ovum, right? And from the mom, and then going to be fertilized by a sperm carrying a dominant allele from dad. So you are going to look at what's on top of this square, right? And then what's on the left from the dad. You're going to write this. So then this is another scenario. The offspring will be heterozygous. Then if you look at this square, again, you look up, get the oocyte from mom, this type of oocyte from mom, and this type of sperm from dad. So you also end with a heterozygous genotype. And then finally, in the last square, so this is a 2x2 table, right? So the last cell, you are going to get a recessive allele from mom, a recessive allele from dad. You end up with homozygous recessive allele. So those are the four possibilities, right, for the offspring. So each one of them you have 25% of them getting this. Each square represent 25% of chance, right? So you have 25% of get homozygous dominant allele, 25% of getting homozygous the recessive allele, right? Then 25% plus 25%, both are heterozygous, then together 50% chance of getting a heterozygous genotype. That's genotype. Then how about phenotype? Phenotype, then if you look at the phenotype, all three, right, homozygous dominant as well as heterozygous present with widow's peak. That's the phenotype. Then the straight hairline is the phenotype, and it's only going to be result from 25% of the offspring. The other 75% of the offspring have the same phenotype, right? But even though they could be with different genotype. So this is what we call square. That based on mom and dad's genetic information, we will be able to get all the combination of the offspring for their genotype. Right, this is for genotype only. But then, depending on what kind of allele that is, then you can base this and decide on what's the percentage of the particular phenotype presented in the offspring. So basically, all the rest of the exercise 12 is giving you different kind of scenario, and you need to be able to generate, to be able to fill out the Punnett square and to figure out what's the percentage of genotype, a particular genotype, or what's the percentage of particular phenotype. And then we are going to first look at some exercise together, but then you should be able to then finish all the exercise yourself. First, let's look at how to write out of the Punnett square, right? So now I'm giving you empty ones, letting you walk you through step by step how to approach that, right? So you are always going to be given genetic information from the mother and father. So for this particular case, right, we're talking about a dimple or no dimple kind of phenotype, and that dimple is the dominant allele. So that's this particular dad, right, is homozygous for the dominant allele. That's capital D, and that's shown here. So therefore, the possible sperms type are all capital D, right? So you can write it here, one D, capital D here, capital D here. And then how about mom? Mom, we always write mom's genetic traits, right? The gametes from the mom on top as the first row. And the mom happens to be homozygous recessive allele carriers, so it's lowercase d. And if you look at then what are the possibility of the possible ovum from mom, then will be a lowercase d here, a lowercase d there. So showing you exact one is to fill up those top row with mom's possible oval type, and the first column, right? Basically, those are outside of those 2x2 table. On the left of this 2x2 table, then write the possible sperm type from dad. And then what they are going to do is to figure out then using mom and dad's gametes, right, already listed here, and look at different combination of them, right? Then for the first square, right, 25%, you take the gamete from dad and the gamete from mom, and then you're going to get the genetic makeup of the first possibility, right? That's counted for 25% because it's 1 out of 4. And so that's a heterozygous for this particular case, right? Based on the mom and dad's genetic makeup, you're going to get a heterozygous genotype for this particular combination. And for this one, then you can look at the second square, right? You can also get a capital D, right, dominant allele from dad, a recessive allele from mom, and then you also end up a heterozygous possibility, right? This possibility also a heterozygous genotype. Then you finish there, the same thing, right? And with a capital D, right, a capital D from dad and lowercase d from mom, and the same thing for the last one. So for this particular type of genotype of mom and dad, you end up with all 4 cells all 4 of them end up with a heterozygous, right? So then if you then try to fill out the blanks here, what's the percentage of offspring has the genotype of homozygous capital D? 0. None of them. What's the percentage of getting a heterozygous combination? Then it's 100%, right? And what's the percentage of getting a homozygous recessive allele? 0, right? For this particular mom and dad genotype, all of the offspring is going to be end up having heterozygous, right? And then this is only look at genotype, right? Then if you look at the phenotype, then because this dimple or no dimple is actually a strict dominance pattern, right? So that means if it has a capital D, then it will have a dimple. Doesn't matter whether it's heterozygous or homozygous. So the heterozygous is going to behave phenotypically the same as homozygous capital D, and then you can homozygous dimple, then basically then phenotypic wise, then all of the phenotypes are going to have dimples, right? Because all of them are heterozygous. Then how about no dimples? Well, that's 0, because no dimples is only going to present if the genotype is homozygous recessive allele. Right. So number one, how do you tell the percentage of the offspring have a particular genotype? You have to be able to fill out the Punnett square based on the dad and mom and dad's genotype, right? And then from the Punnett square, figure out the type. Then looking at the inheritance pattern, right? If it's dominance, then you just follow that the phenotype. Well, what if the inheritance pattern is not strict dominant, right? So you guys in the exercise in page 3, right? Exercise 12, page 3. Then it's talk about a scenario that you do not have complete dominance. It's the if it's the heterozygous going to present with a phenotype that's intermediate between the homozygous dominant and homozygous recessive. So that means the dominant allele is not completely dominant. So if you only have one copy of that, it's actually intermediate. So the results, the example we give you is the straight hair versus a curly hair, right? So the straight hair is the dominant allele, the curly hair is a recessive allele. But when you have heterozygous genotype, that means one copy of the straight hair, one copy of the curly hair, and then heterozygous, you end up with wavy hair. And so how do you write up the Punnett square is still exactly the same thing as if you were doing this. But then, and even for the percentage of chance of getting the genotype is the same as if you are doing this, because you basically just count the squares. And each square is going to count for 25%, and so you can count how many squares you end up having. Then for a particular genotype, then you can figure out the percentage of chance offspring have a particular genotype. Only when try to figure out the phenotype, then because of the inheritance, the phenotype presenting is going to be, the incomplete dominant type of phenotypes. So then the heterozygous is going to end up with a third phenotype. It's not only dominant phenotype or the recessive phenotype, but then you have an intermediate phenotype. So, that's, I'm not going to go through how you are going to do that, because it's the same thing, just when you figure out the phenotype is slightly different. But I'm going to talk a little bit more about the co-dominant pattern. Okay, so this is a pattern, then it's represented by ABO blood type. I know when we talk about ABO blood type, some of you take a while for you to understand, but here it's kind of showing you why. Okay, so the ABO phenotype is co-dominance. Basically means that you have 2 dominant alleles, not 1. Okay, so instead of 1 dominant allele, you have 2 dominant alleles. Then if both of them are presented, then you are going to end up with, a different phenotype. So then you have more, phenotypes than a straight, 2 allele, kind of 1 dominant allele and 1 recessive allele type of pattern. So let's look at this. The phenotype, right? The antigen A or blood, the antigen A or antigen B on the red blood cells are encoded by this dominant allele A or B, right? If it doesn't, encode any antigen, so that will be a 0. It will be a O, right? So giving you an example, right? If dad's genotype is AO, and then what's the phenotype? That should be, because A is dominant, then the phenotype should be A. The father is type A. And the mother's genotype is BO. The mother's phenotype should be B, type B, because B is also dominant. Right? If you write up, right, mom on top of the 2x2, that's mom's gametes. That's the two type of allele, two alleles from mom. And on the first on left side of the Punnett square, you have dad's two alleles, A and O here. Right, after you write it up like this, and then you can look at for the first square, that you're going to get one allele from dad, one allele from mom, you end up with AB, right? Then for the second square, you're going to have one allele from dad, one allele from mom, so you're going to end up with AO. Then this one will be BO, right? And then the last one will be OO. Okay, so you can write up of the percentage of chance the offspring have the genotype, right? The genotype for each one of those are going to be 25%, because each one of them going to be only from one square. And then how about the phenotype? Well, we said AO is A, BO is B, right? Because A and B are both dominant allele, but then AB is going to have a blood type AB. And the homozygous recessive allele, then you are going to have a blood type O. Okay, so that's why from mom and B type E, another has a type A, you could end up with, equally chance of getting offspring with each one of the blood types. Okay, so that's kind of how you still do the same. We still do the same thing of using the Punnett square to figure out the genotype. Then it's only the phenotypes is slightly different again, because the co-dominance pattern. Okay, lastly, we're going to actually look at the sex-linked inheritance. And mainly we're going to look at the X-linked, right, a recessive inheritance. Okay, so this is the part that your page 6, 7, 8, all going to devote of the exercise 12, all going to devote it into this X-linked inheritance. So X-linked inheritance, because X chromosome is much bigger than Y, right? So here, if this gene is on the X chromosomes, chances are it's not going to be on the Y chromosomes, because Y chromosomes is very small, and the supermajority of the Y chromosome genes are actually related to the sex, traits. So male sex trait. So X, chromosomes gene doesn't really have anything to do with the sex trait. So if it's on the X chromosome, chances are it's only on the X chromosome, not on the Y on the Y chromosomes. Okay, so that makes it even more complicated, right? Because then the male and females are going to govern by different rules. Okay, so let's say, using example, right? We now have to not only write up the alleles, but also write up of the sex chromosomes. So you are going to be write up. So if the mom genotype is one of the X chromosome carry a dominant allele, another one carry a recessive allele. This particular trait is as color blindness. Okay, so then it's indicated by a capital letter C. So that is actually the normal allele, and the lowercase c is the colorblind allele, because that's a recessive trait. Okay, so the mom carries a copy of the color blinded allele, recessive allele. But the mom, because it has another X chromosome carry a normal allele, the mom is not going to have any phenotype. Doesn't have color blindness. It's only a carrier. But then the two type of gametes that are going to be generated by the mom is going to be written up like this. And so now we have to include the X chromosome meaning. So XC or XC lowercase, that's the 2 possible X chromosomes that are coming out from mom, right? And then from that, then because that only have one X, and that is actually a normal colorblind gene which is dominant, then you write up the Y, the gametes for the sex chromosome from that is 50% of them going to be X chromosome, carry a normal allele, and 50% of them going to be a Y chromosome. So then you again look at the combination. You get 1 gametes from mom, 1 from dad, and you end up with 2 copy of X on the top row, right? Those are all female offsprings, and then the bottom row is going to get a copy of Y chromosome from dad. So it's all going to be male offsprings. So if you look at this, right, then you write up the chances of the offspring have a particular type of genotype. You again count 25%, 25%, right? You can write up like this. But then when you get to the phenotype, right? So you have the female normal vision that would be this, right? So 2 copy of the normal allele, and the female carrier also is going to be having female version. Is going to be female version. Vision, right? And then so then the males, you are going to have normal 25% of the males going to have normal. 25% of the offspring is going to be male and also normal vision, and then 25% of them going to be male, then have color blindness. You can see none of the females have color blindness, but then half of the male offspring is going to have color blindness. So the daughters from this kind of mom and dad, none of them going to present, right, with color blindness, even though half of them are going to carry the genetic, the color blindness allele, but then they're all going to be normal vision. And then it's just the males, the sons from this couple is going to be predominantly affected. The female is going to carry this recessive allele to the next generation. It's going to influence the grandson from the daughters. But for the X-linked chromosomes, so your inheritance for your exercise basically listed all different types of scenario, right? Whether it's carrier female, affected male, or normal unaffected female. So it's going to be you're going to have a lot of, I think, 6 Punnett square to work on, and then for each one of them, going to figure out individual their genetic makeup and their phenotypic makeup. I'm going to upload my a key for this, for the exercise 12. So then you guys can first try to work on the Punnett square yourself, but then you can compare to know, compare that to my key and to know whether you got it right or not. Hopefully that's going to be helpful for you. For the exam, expect to have some type of exercise like this. Maybe not having you write up the Punnett square, but you may actually have to still choose, what's the phenotype or the genotype of particular type of couple, for a particular type of scenario. So be real familiar with what, this, how to, based on, parents' genotype, figure out offspring's genotype, and based on particular genotype, have to figure out their phenotype, right? So expect to have those kind of questions in the exam. That's all for our last lab.